Okay, hello everyone. So basically, today's topic is complementary genes. Okay, so we will understand about the complementary genes. To understand about the complementary genes, first of all, you see, you have to understand that complementary genes are two such genes. Complementary genes are two or three or more such genes which will produce a single phenotype when both are present in dominant condition. Both are present in dominant condition. When both the genes are present in dominant condition, both giving rise to a single phenotype. Okay, don't please confuse it with pleiotropy. In pleiotropy, one single genetic location were controlling different phenotypes. Here, it is just opposite of pleiotropy, where the single phenotype is controlled by two or more genes located in two different loci of two different chromosomes. Like for example, located both the genes located in two different loci of two different homologous chromosomes. Okay, so let us try to understand the definition and whatever I have said here. To understand the complementary genes, the definition, we will take the example of Lathyrus odoratus, which is actually sweet pea, not sweet potato, sweet pea. Okay, now this Lathyrus odoratus sweet pea, its flower color can be either purple or white. Now, what happens is, at first there is a presence of raw material which makes the color of the flower white. If the raw material is converted into chromagen due to an enzyme E1, still the flower color will be white. And if the chromagen with the help of an enzyme 2 is converted into an anthocyanin, still the flower color will be in this case the flower color will be purple. So what did I say? I said that if there is a raw material present, there will be no chromagen produced and the flower color will be white. If chromagen is produced, still the flower color will be white. And if from chromagen anthocyanin is produced in the cells, then the flower color will be purple. From the raw material to chromagen conversion, responsible is enzyme 1, and from chromagen to anthocyanin conversion, responsible is enzyme 2. Now, enzyme 1 is actually produced from the gene C, and enzyme 2 is actually produced from the gene P. Okay, so it is produced from gene C and gene P. Now, what actually happens here is that, you see, suppose two pairs of homologous chromosome is there. Okay, now let the C be located in the dominant form in this loci and in the recessive form this loci. So the allele is capital C, small c. It could be capital C, or it could be small c, small c. These three possibilities are there. Okay, fine. Now in the another locus, it can be capital P, small p, or it could be capital P, or small p, or capital P, small p, as depicted in the diagram. Now the fact is this: in the dominant form, the enzyme will be produced. In the recessive form, be it in the homozygous dominant form, still the enzyme 2 will be produced. Be in the heterozygous dominant form, still the enzyme 2 will be produced. But if it is in PP form here, small p form, then the enzyme 2 will not be produced. Keep that in mind. In the same way, in the capital C, small c form, the enzyme 1 will be produced, because from this and then in capital C form also the enzyme 1 will be produced, but in small c condition the enzyme 1 will not be produced. So if I take it in this way, capital C, small p, capital C, small c means in this condition, then purple flower will be produced because from here chromogen will be produced due to this gene anthocyanin will be produced. Here also purple color because these are this homozygous dominant situation. In this case it will be white. Small c means at first the small p, that means what? Enzyme 2 will not be produced. Doesn't matter. Small c is there. That means enzyme 1 will not only be produced. So raw material will remain like that. So white coloration will be there. If it is small c, capital P, you will expect that enzyme 2 is produced, so anthocyanin is produced and the flower becomes purple. But no, you can see the combination is small c. That means the enzyme 1 is not produced and its precursor chromogen is not produced. So raw material will be produced, will be there, and the flower color will be white. Okay, and in the case big C and small p, it will be white because only the in the small c capital C condition the chromogen will be produced which will give the flower color white, but then the anthocyanin will not be produced, so purple coloration will not be there. However, there can be other combinations also, we will understand. I hope that you have understood up to this much. If you have understood up to this much, now let us try to understand the complementary genes. Now I hope that you have understood that in complementary genes what is happening, the gene C and the gene P are complementary to each other, which when present in only dominant form, either heterozygous or homozygous, produces a particular phenotype purple. If they are not present in dominant form, both of them not present in dominant form will not give the purple phenotype. One of them present in dominant form, one in recessive form, still purple phenotype will not come. Okay, so my definition that both of them should be in dominant form, either heterozygous or homozygous, to give rise to a particular phenotype, that is purple coloration in this case, is the definition of complementary genes. And both the genes, it is a kind of intergenic interaction, if I can say. This is a kind of intergenic interaction. Now in case of intergenic interaction, what happening that two genes located at different loci of different chromosomes interacting with each other to give rise to a particular phenotype will be known as complementary genes. I hope that you have understood up to this much. Now moving, let us try to understand its heredity. You see, let us first do it in the style of Mendel. So if we try to do it in the style of Mendel, how can we do it? Let's see. Hold on. Okay. So just I'm so sorry. Yeah. I hope that everything is fine now. Now, what actually happens is, suppose let us take the parental P1 generation to be capital C, capital P, homozygous dominant of both the genes, crossed with a small p. So this will give purple colored flower and this will give white colored flower. Now what actually will happen is that this will produce its gametes, and its gametes will always be CP combined with the gametes small c, small p. So in each of the cases you will get a F1 generation of CCPP, which will give rise to purple coloration. I hope that you understand this. This is not a very big deal to understand, because if there is, according to Mendel, there is segregation or independent assortment, whatever, then C will come with that, P will come, or C will come with P will come, or whatever combination. So these are the gametes which will combine in the F1 generation to give rise to this. Now let us fertilize cross the CC, they do the self fertilization of them crossed with CC PP. Now what will be the gametes produced if we do it in a Punnett square? 1 2 3 4, 1 2 3 4. Now the gametes will be CP, okay, capital C small p, okay, small c capital P, okay, and small c small p. Fine. And here also the gametes will be same of the male or female. Now what happens is this will be CC PP, CC PP, CC PP, and CC PP. Okay. In this case it will be CC capital P small p, this is small p, capital P small p capital, I hope it is understandable. So again capital C small p, capital C small c capital P small p, capital C small p, capital C small c capital P, capital C capital C small c capital C small c capital P small p, and small c capital P, and it will be small c capital P small p, capital C small c capital P small p, and capital C small p, and small c capital P small p, and small c small p. Now this will be both in dominant condition, purple P, purple PR, purple. Chromagen, see, chromagen will be produced, anthocyanin will be produced. Chromagen will be produced, anthocyanin will be produced, purple. Chromagen will be produced, anthocyanin will be produced, purple. Chromagen will be produced, anthocyanin will be produced, purple. Chromagen will be produced, anthocyanin will be produced, purple. Chromagen will be produced, anthocyanin will not be produced, white. Chromagen will be produced, anthocyanin will be produced, purple. Chromagen will be produced, anthocyanin will not be produced, white. Chromagen will be produced, anthocyanin will be produced, purple. Chromagen will be produced, anthocyanin will be produced, purple. Both in dominant condition, heterozygous, homozygous. One is recessive, another in dominant, white. Because chromagen will not be produced only. Chromagen will not be produced only, white. Purple. Okay, so white. Because their chromagen will be produced, which will give rise to white, purple. Small p, anthocyanin will not be produced. Chromagen will not only be produced, raw material is there, so only white and white. So 1 2 3 4 5 6 7 8 9 9 purple, 16 are there, so 7 1 2 3 4 5 6 7 white. So what actually has happened, the ratio changed into 9:7. If there is a normal Mendelian cross, 9:3:3:1 would be your ratio, right, where all will be dominant here, one dominant, one recessive, the other dominant, other recessive, and all recessive. This was your, if you remember my previous teachings, this was your thing. But here all the dominant will come, this 9 will come as 9, but this 3 plus 1, one dominant, one recessive. If one of them become dominant, other becomes recessive, still it will give white flower. the other dominant, other recessive, still it will give white color, and it will also give white color. So being present in both dominant form, which is 9, will give rise to purple color, complementary genes. But if one of them is present in recessive form, then there will be white coloration. So this 3 plus 3 plus 1 will give rise to 7. I hope that you have understood about the complementary genes. So this is your details of complementary genes.